Hello everyone, thanks for clicking this video and your interest in using our platform. In this video, I will give you a brief introduction and a demonstration of the usage of our online single-cell analysis platform, GranatumX. GranatumX is a community-engaging, modularized, and flexible software environment for single-cell analysis. The goal of the GranatumX is to provide biologists a better access to bioinformatics tools and the ability to conduct single-cell data analysis independently without prior knowledge in any programming language. Of course, for bioinformaticians who are interested in embedding their single-cell RNA-seq specific method within our platform and pipeline, they will be able to do so through the Gbox configurations. For more details on that aspect, please refer to our preprint here. The link is listed here. I will not talk in details about this in this video. In our platform, we offer a default analytical pipeline as listed here. We expect the user to have the single-cell RNA-seq count matrix compiled from any software or experimental platforms. In addition, you could provide any metadata that describes your experiment, such as tissue type, locations, or treatment conditions on each individual samples. After you upload your files, you could perform single-cell RNA-seq imputation that is backed by our own developed imputation method, deep impute. Then you could proceed analysis with common preprocessing steps such as cell normalization, gene filtering for highly variable genes, and log transformation of the expression value. In order to visualize your high-dimensional single-cell RNA-seq data, our platform provides popular methods such as linear dimensional reduction method, principal component analysis, or nonlinear dimensional reduction method such as UMAP visualization or t-SNE. Now that you have QC'd your cells, normalize them, and determine the relevant visualization coordinates, we can then proceed to determine cell clusters and perform other downstream analysis such as marker gene identifications while differential gene expression analysis. One advantage of GranatumX over other platforms is the ability to configure your analytical pipelines. That being said, you could add or delete any steps or methods that may or may not fit best for your analytical purpose. In addition, you could always reorder your analytical step anytime. In this video, I will give a brief lab demonstration of how to use our platform using this example dataset. This dataset is reported by this manuscript called Application of Single-Cell RNA-seq Sequencing in Optimizing a Combinatory Therapeutic Strategy in Metastatic Renal Cell Carcinoma. In this example dataset, we have a count matrix with 180 cells and 90,924 genes. For metadata, we extracted information such as state of cancer, primary versus metastatic, and sample type, parental tumors, or patient-derived xenograft, etc. Now let's go to the lab demonstration. First, you want to put in this URL address that can be found at the beginning of the video to go to the GranatumX application page. From here, you will find useful information regarding browser requirement and matrix preparations. Most importantly, you will find the link to the online platform of GranatumX. By clicking this link, it will take you to the main GranatumX page. For this demonstration, I will use the default comprehensive pipeline. A couple of remarks here. First, you may want to change the project name into something that describes your analytical purpose. So for here, I'm just gonna name my project as demo. And also, the second thing is that before you perform any downstream analysis, you may always want to record this sandbox ID. Because this will allow you to recover all your analysis if you are experiencing any network failure. In addition, you can share your project with another people using the sandbox ID. Now let's start this analysis. The example dataset that I mentioned earlier can be downloaded from here by clicking those two links. However, since I already have them in my computer, I will just gonna go ahead and upload them. You will need to make sure that the uploaded data type matches with the argument here. So for both of my assays and metadata, it will be CSV file. And you want to make sure the species is human. So just another remark is that if you are dealing with a large data set, you may want to choose to zip file your assays to achieve a quicker upload of your data set. So this step normally won't take long, but if the page sometimes showing you running without any progress, you may just want to consider to refresh your browsers, as most of the uploads won't take long. After the uploading process complete, you will be able to see a brief descriptions of your uploaded dataset, such as the dimensionality of your data and the corresponding metadata that you uploaded. So in our pipeline, we actually provide an imputation options backend by our own developed tool DeepImpute, which is a neural network-based imputation algorithm. For more details regarding this method, please refer to our published manuscript. We will use all default settings here for these imputations, and for the following analysis, if I'm not specifically mentioned, it will all be default setting. So the DeepImpute step may take a while, so you may want to wait patiently for this step. So after the imputation process completed, you will be able to see the results as a form of heat map to demonstrate the dropout frequency change before and after imputation. Also, there's numeric measures for the imputation qualities. For example, the rate of the dropout before the imputations is around 58%, and the rate after is around 12%. After imputations, we could then move to more common preprocessing steps such as cell normalization, gene filtering, and log transformations. So for normalizations, the default method is quantile normalization that will eliminate unwanted variations between the cells caused by different library size or different data distribution. So to make the plot prettier, I'm gonna click the box to log transform the box plot with pseudo count of 1 added in the expression value. So here are two box plots demonstrate normalization qualities for randomly chosen 40 cells before and after normalization. Each bar here represent one cells, and you can see that for this very skewed cell distribution, it got corrected after the normalization. So gene filtering step is backended by Scampi, a Python-based single-cell RNA-seq package. This step will remove genes that are expressed too low using the count-based filtering and extract genes that are highly variable across the cells using dispersion-based filtering. So a couple of past publications has demonstrated that it is very preferable to use the highly variable genes to perform downstream analysis, such as dimension reductions and clustering. Again, we're using default parameters. So here are two plots. I'm gonna zoom in a little bit. So here are two plots demonstrate the filtering result for all the genes. Each dot represents a gene with x-axis being the mean log transformed expression of these genes and the y-axis being the dispersion of the genes. The gray dot here are removed genes based on the default threshold that we're setting here. And you can see that in the end, we have 5,131 highly variable genes extracted from the original gene set. And log transformation is normally performed before the downstream analysis, such as clustering and differential expression analysis. The reason is that for single-cell RNA-seq expression, normally follows a negative binomial distribution with which is skewed heavily towards the low values. And this skewness may affect some statistical analysis that assume a normal distribution. So here we're using the log function of base 2 and a pseudo count of 1. So those two histogram here shows the distribution of the expression level before and after log transformations. And we see how log transform actually correct this highly skewed distribution to the one that we observed here. And for high dimensional data, the common practice is to apply dimension reduction method to calculate the low dimension embeddings of the input assays using either linear or nonlinear dimension reduction method. So here we're providing PCA, t-SNE, and UMAP to visualize cells on the 2D plane. You could actually perform all these three dimension reduction methods in parallel using the log transformed assay. So both of them, we're gonna just use, for all of them, we're just gonna use the default settings. So here is the PCA plot, t-SNE plot, and the UMAP plot that generated. It is actually more common in the field to use the UMAP coordinates. So we're gonna explore in detail about this plot here. So in this UMAP plot, we can roughly detect two clusters that are relatively separable. So in order to detect the true underlying subpopulations within these assays, we need to perform unsupervised clustering to group cells that are biologically similar. So in the default settings that we're gonna use the Lewin graph clustering method backended by the ScanB. So this clustering method is based on modularity optimizations. Again, we will use the UMAP coordinates. So this results gives us three clusters, and recall that back into the file uploading step, we actually include some sample descriptions, aka the metadata for each of the cells. So those metadata actually can be superimposed onto this sample scatter plot to help you get an idea of why those cells got categorized together. So we're gonna go to the sample coloring step, and we're gonna choose the UMAP coordinate, and the value that we want to superimpose is gonna be the type from the metadata that I uploaded. And you want to so for the sample recovery step, we can both choose the continuous metadata values or categorical metadata values. So you want to make sure this is correct. So if we're gonna explore this coloring patterns, we can tell that those three clusters identified in the previous step is actually represents three types of cancer samples. So this is the primary cancer from the patient-derived xenograft models, and those two clusters are all metastatic cancers, but with one from the PDX and one from the parental tumor. So we can see that the clustering algorithm actually groups cells into different type of cancers correctly if we look back to the clustering assignment. So now that we have determined the cell clusters, GranatumX actually provide a common downstream analysis such as marker gene identification, gene set enrichment analysis, and pseudotime construction. For marker gene identification, the default method here is t-test, and you can find the globally differentially expressed genes for a certain group. You could actually customize which group vectors that you want to use. It could be either cluster assignment from the clustering result or the metadata that you provided. So for the demonstration purpose, I'm just going to use the type because it's pretty much the same as the clustering assignment from our previous inspection. So the marker gene identification result will show you a graph that can order the genes by their rank and show you the highly differentiable genes from each group compared to the rest. You can actually download those results here as CSV files just by clicking here, download. I already downloaded them in my computer, so I'm gonna show you what the file's gonna look like. So it will be just gonna be a list of genes that ranked by the score. So by using these results from the marker gene identification step, you can actually then perform the gene set enrichment analysis to find biological process or pathways that are enriched in this group. So please be sure to, when you're using this step, you want to make sure the species is correct, and you want to choose the desired database. It could be gene ontology or CAG. So in my analysis, I'm just going to use Go, and I'm going to detect the PDX meta versus the rest of the cells. So this step is going to take a while, so let's just going to jump to the pseudotime construction. So just another remark. So during your analysis with GranatumX, you can always, when you're waiting for the result from one step, you can just jump to another one and start do the analysis at the same time. So for the pseudotime construction, it is assumed that the temporal order of the differentiating cells is encoded in their single cell expression profile. So the default method to estimate this order is in here is using the diffusion pseudotime, which measures the transitions between cells using diffusion-like random walk. So this implementation will make it possible to reconstruct the developmental progressions of the cells and identify transient or metastatic state of the samples. Or you can also find out the branching decisions and the differential endpoints from this pseudotime construction result. So here I'm gonna using the default setting. So both steps may take a while. So sometimes you just need to be patient to wait for the result. So while we're waiting, actually just gonna set that, as I mentioned earlier, the GranatumX is actually very flexible in terms of orders and steps. You could always add or delete a step freely in this pipeline by using the plus and X here. So you could also change the order by dragging around this step to move your analytical step from one place to another. Of course, it will just gonna be fit with your analytical purpose. So for instance, I could actually just add a step here after the sample coloring. If you click on plus, you will actually find a list of available methods that can be added and also available now in this in our GranatumX platform. So for the demonstration purpose, I'm just gonna do a color scatter plot. It is mentioned in the paper that for the metastatic samples, it will have high EMT signature. So let's search for a very signature EMT genes such as WIM. And for here, you may want to choose the normalized assay to include as many genes as possible. And I'm gonna click submit. Of course, you can actually do multiple genes here to overlay on top of each other. For here, I'm just gonna use one. And as you can see, and if you want to go back and see this sample scatter plot, you can see that the VIM expression is actually mostly highly expressed in the parental tumor-derived metastatic samples. So by using this step, it will actually allow you to explore more on your single cell data. So now we have the gene set enrichment analysis result ready. Let's take a look at it. It will just gonna be a table with a list of gene set names, and it have p-values and false discovery rate, and it will be ordered by how much they enriched in this group. And for these tables, again, you can download it as CSV file. And once you download it, it will just be a table that has all the gene set names, ordered gene set names, with all the statistical values here. So for the SUTOM construction results, so let's take a look at the output here. We actually have two plots. So for DC1 and the DC2 axis, it actually indicates the values of the first two diffusion components. They're the top two largest eigenvalues of the transition matrix, which approximate the dynamic transitions of cells through stages of differentiation process. Just like UMAP and PCA plot, DC1 and DC2 here is used for visualizing a single cell in the two-dimensional plot. The differences between the two output plot is about their color keys. The first one is a continuous color keys, and the second one is the discrete color keys, both of which can be used to show the differential stage of the cells. So if a cells here, for instance, if a cells here in the first plot has a color that corresponding to a smaller values, for instance here, it actually means the cell differentiate early, while the one with the color keys that corresponding to a high value, for instance here, it actually represents cells that differentiate late. As to the second plot, the continuous time values is actually just be discretized, and the cells are correspondingly divided into different groups according to their continuous color values. So the cells in the group with smaller color values, for instance here, is actually being grouped into one, as we can see is grouped into group 0. That indicate a cell that differentiate early. So that pretty much sums up the analytical step in the GranatumX default analytical pipeline. So at end of your analysis, you could generate a report by clicking the project report here. It will give you a page with all the analytical step and results, the figures that you had from this pipeline. So you can always you can save it as PDF file by click the Control P to save as PDF for easy storage. So that is the end of the live demonstration. Our platform is still under active improvement, and if you're experiencing any technical issues, please contact the GranatumX team here using this email address. We'll welcome all kinds of suggestions for the purpose of improvement. And thanks again for your attention and your interest in our platform.